Welcome, everyone. Listen, if you happen to be a microscopic organism, you're going to want to pay close attention because consider this your ultimate survival guide. Today's explainer isn't just some boring list of lab protocols. Oh, no. It's a harrowing look at the rigorous daily hazards we microbes face in a scientific laboratory. Now, from a human perspective, sure, these are just standard procedures for the safe cultivation and study of microorganisms. But from the perspective of a bacterium, this is a terrifying gauntlet of total annihilation.
Okay, let's dive into this map of danger zones every microbe has to navigate. We'll start with the principle of sterilization, move into extreme heat, explore rays and filters, and wrap up with chemical methods. So, Section 1, the principle of sterilization.
So, the absolutely crucial point here is that sterilization leaves no survivors. None. It's the process of making an article, surface, or medium free from any type of microorganism. And human scientists demand this incredibly strict standard because, even one single rogue bacterium can ruin an entire experiment. But for us microbes, this definition literally spells doom. There is no hiding. Every kind of microorganism is wiped out.
Now, you might hear the word disinfection and think, oh, that's basically the same thing, right? Well, not quite. Say you're a microbe just hanging out on a lab table. You might face disinfection. That means getting wiped out by a glycolic acid compound or carbolic acid. Disinfection is all about reducing microbes on objects and surfaces, but it is not used on culture media. Sterilization, on the other hand, is absolute obliteration. It's the uncompromising standard reserved for the actual culture media the scientists are working with. And speaking of absolute obliteration, just picture incineration, also known as flame sterilization. Imagine you're just clinging to the surface of a bacteriological needle. Out of nowhere, that needle is thrust directly into the open, roaring flame of a Bunsen burner or a spirit lamp. And the human just holds it there until the metal literally becomes red hot. Everything on that surface is instantly destroyed. It is a terrifying, fiery end. Let's move to and see how this builds in Section 2. Physical methods, extreme heat. Because as it turns out, open flame isn't the only weapon humans use against us. There are two main extreme heat environments you gotta watch out for: moist heat and dry heat. If you're hiding in water or in culture media, you're gonna face moist heat. Scientists use steam under pressure to sterilize these liquids, usually throwing them into a pressure cooker or a heavy-duty industrial machine called an autoclave. It is literally a boiling nightmare. But on the flip side, if you're lingering on glassware, you're gonna face dry heat, which comes from a hot air oven. And just how hot does that dry heat oven actually get? 150 degrees Celsius. Yeah, just hearing that number should make any bacterium shiver. If you're residing on a glass syringe, forceps, a scalpel, a pipette, a flask, or a petri dish, you're locked in this scorching oven at 150 degrees Celsius for a full 30 minutes. Absolutely nothing survives that kind of sustained, blistering heat. Section 3, physical methods, rays and filters. Now, what if the environment isn't hot? Well, don't let your guard down, because the lab is absolutely full of unseen dangers. Before scientists even sit down to work, they deploy this invisible threat: ultraviolet radiation. They use UV lamps to flood inoculation chambers and lamina airflow stations for a solid half hour before they do anything. It just fries any microbes lingering in the air or on surfaces. And interestingly, these UV lamps have to be enclosed in quartz. Why? Because regular glass won't actually transmit UV radiation. It's a brilliantly deadly little workaround. And this brilliantly illustrates what happens when scientists can't use heat because, certain substances like enzymes, antibiotics, amino acids, and vitamins are heat sensitive. They'd be completely destroyed in an oven or an autoclave. So scientists use an agonizing trap instead: membrane filtration. They place a millipore membrane filter inside an assembly, autoclave the whole assembly first to make sure it's deadly clean, and then use negative pressure or suction to pull the solution right through. The filter retains all the bacteria, and what passes through into the sterile container is a completely microbe-free filtrate. Which brings us to Section 4, chemical methods of sterilization. So if the extreme heat, the invisible radiation, and the microscopic traps weren't enough to worry about, we now arrive at the toxic liquid and gas chemical arsenal. Now, what's really interesting about this is how methodically scientists match the specific chemical weapon to the environment. Look at the alcohols. They use 70% ethanol or 70% isopropanol just to casually wipe down working tabletops and inoculation chambers. Then there are the aldehydes. If the number of microbial contaminants gets a little too high in the lab, scientists won't just wipe the tables. They'll go ahead and fumigate the entire laboratory using formaldehyde gas. And finally, we have the inorganic chemicals, specifically heavy metals and hypochlorites. I mean, these are highly toxic to any organism. Scientists use solutions of copper salts or mercury salts. Take HgCl2, for example. At a 0.1% or 2% solution, it's a super common disinfectant used directly on seeds, plant tissues, and other biological materials. They also bring in the heavy hitters like a 10% solution of sodium hypochlorite or calcium hypochlorite, applying them for a brutal 5 to 10 minutes just to guarantee absolutely nothing survives. When you step back and look at the sheer scale of these methods, flames that turn needles red hot, pressure cookers, 150-degree ovens, invisible quartz-filtered radiation, microscopic suction traps, formaldehyde gas, and toxic heavy metals, it really leaves you with a profound thought. With all of this standing in their way, the fact that microorganisms continue to thrive, adapt, and wildly outnumber us on this planet is a true testament to their incredible resilience. It really makes you wonder, just how tough are the microbes that manage to survive in the wild?